You got it. I love it. Certainly. Respected, my director, in fact, is our director, most of our students will go back at it. And we could chairperson, Madam, seriously, we have posted release and also the universe and my dear colleagues. I have other performance studies here, so probably in the last as a scientist. So thank you for inviting me. First, I apologize. I had a bad throat. Last week, days, there were some people before. So, what do you use my acceptable? I studied all my education from Canada, and basically from Congo, I should have the son of the soil. I'm always happy with all my school education, looking from the soil only. Not even if you want to. Second thing I was saying is a better manager for a long period. I was working at virus and the post infection and the producing system, these are all the previous studies. I mean, then I moved to seed organization. I thought better my question should be it should be relevant to the seed organization where I up to be. This February is a very serious problem. Now we have recently also been had a problem there. We have serious, rigorous screening of disease. So whatever we are having, it is unlike like Eri or Muga or Tasa, we can compromise the rest of different transport. But whereas in Mori, many different research is there. We can compromise, we have to eliminate or we have to make appropriate disease management to control the disease. The Louis Pasteur identified as a mother examination for hundreds of years, which are going to do that. But we could not still replace what are the Louis Pasteur. So, what are the modern strategy? But where we are now? What would be the next new model? What we is very brief, that were minutes, which I am not going to discuss the part of my job, how we express the protein, how we do it. Now, see, 2000 years back, they played like and Peruvian factor. And I'm going to talk about people. This is a proper disease management. Nodi Nadi, Mundal Nadi, other than other Nadi by which makes people want to do anything if you want to control the disease. Identify the population. You please take care proper medical, how to remove the or how to prevent the infection. Then only to do it. So this is the best thing. 2000 years he has identified. But still we could be able to do the same switching. Today our subject of the presentation is this. This is the Nosema analysis spore. It's a very special spore. We will be thinking when the bacteria is there, this is a host cell. When the host cell is infecting the bacteria is infecting that, bacteria infect multiple inside the host cell. But the Nosema bombices is a special mechanism. It is a host cell. It has got a symbol tube. One host cell is there. This Nosema bombices will go and sit on there. This is like a big injection. It will inject the inside the host cell, releases the nucleus inside. So it has got a big injection procedure there. It's a very long. So it's a special provision there. And this pathogen never enter inside the host cell. Only uses the product to inject the nuclear material. So India is the second largest producer. So I don't want to tell you here, most of us are productions is all the crop loss mostly by the viruses, fungus, and the bacteria. Now, 40% crop loss. It's a very serious problem. Like as I told you, if anything is there, we have to remove, discard the water. It's not like that. When you ask them to discard it, already since I'm working the seed organization, already the producer seed is infected with February. If I ask the reject it, it's a lakhs of rupees invested has produced the POC. The POC has produced it. We cannot ask them to give it. Or else I will convert them please from effort to commercial because effort is 1,300, commercial 800 rupees. They are also there. It's a big problem. So you cannot do once it is written that you should be vigilant to remove the part from the picture. Please be free from this one. Now, it's a very important thing here I want to tell you. Sorry. When you talk each pathogen, it has got a specific host. If you take a BMNPV, it will infect only the Bombix node. IFO virus, it will infect only the certain specific crops one. But when we talk about the microsporidium, it has more than 50 established host species are there. Nosema bombycis, it will not be Nosema species. Nosema bombycis has more than 40 to 50 host species are there. Bibinia bassiana, just before he was one professor, he was telling that he is using the for the common leptopteran pesticide, biopesticide, that also some host broad host traits. The Nosema, second thing, it is coming from the mother mark to the offspring. We know that. Then the contaminated link, if it is a PD, it will go through the paroral, so it's out. So two type of infection is there. So this has got a broad host and will be the this. It's a basic information we should keep it for the making appropriate disease management. As I told you, this is a spore. See, the God or nature has programmed in such a way when the spore is going inside that rumen, will get sense, can understand the sensors and the when pH is very high, around more than 10 to 12, it bursts. See, this injecting the product tube inside, big coil, this nucleus is coming inside, is a mirogamy, sporoblast, then the sporoblast different, the spores are coming out from this one. For the mother month examination, what I am doing here, once it is the mature spore, if you do the testing, it's a possible. If it is only with intermediate stages, though it is infected with the membrane, you are first to declare this membrane. So we are very sincere. You know how we are NSSO, we do night and day work for membrane testing. Sometimes skips due to this very rather mistake stage we cannot able to find. So we have to develop a diagnostic technique. Not only the mature spore, we should be able to identify this status also. How we have done it, we'll show you. So we first year, from the last week, we got a parallel assessment. Please, we can go through that. The last is hand was so we have got a parallel assessment. See, nowadays, if I have headed, I told you, I have a ear problem, you have to go to this specialist. That gastroenterologist. Heart, I got a pulmonary cardiologist. The cardiologist cannot check the gastroenterology. This scientist, great scientist, is a microbiologist. He's a physician. He was working on the other medical methodology. When the king called, the French Sericulture industry completely collapsed to the Berlin question. He thought every question you are looking at it. Sir, I am working on the pasteurization of the milk. I am going to develop the vaccine for the package log, which I am not going to. But if you think about you please identify the reason what will be the root cause of the disease. Because they are cleaning the room, bringing the fresh water, again the brain disease. They could not appear back from this method. Once the invention is identified, it will be very easy. But getting that invention, getting that information, it's a very difficult, that's a very challenging task. Finally, this, it took the mother mark, checked it, passed it, allowed to lay the egg, and the legs are, that the offsprings are coming up from the infected gravid female, it all having the offspring. So he said, see man, it is coming from the mother mark, not from the female. So we started the mother mark examination, but still it was the reason mother mark that French sericulture industry completely collapsed. It moved to Guma prefecture, to the Japan. They said, you please produce the silk, give it to France. It says everybody know that. The brain is outdoor this one, always pathogen and host are very nicely impacting. And the host is evolved. The pathogen also co-evolved with the host. For example, one thing I tell you, in the CASAR, it's a 6 months diapasm the pupa. When the baby is infected with CASAR mark, 6 months is also the diapasm. So triggering that increase the horses under the diapasm increase slowly. When it's coming out from the mark, it's become very active, the pattern becomes very active, it's going to the next stage. So it's going on with this one. Furthermore, arsenic, hence it's very difficult to control the spread. Once you leave it, sorry, you should tell them, not a you are trying to test only 10 P4 vehicles. P3, please try to call your alarm here. Once you leave it, if it is a P3, sorry, to P2 or P1, that's all, millions. So you have to test it at the early stages to control it. Then, what is the problem with our microscope? It's a time consuming. Sir is posting the many persons from Vijay from Parampur, Tipur, Vijay Hari from Parampur. Come, everybody please come there, madam. You don't know that. Anyway, the favorite problem is that I am going to bring all the people who have there to test it, to make it disease free. It's a time consuming, labor intensive, do night and day. P1 DFOs, so much demand. You cannot test even the Hathamani and Nagatamana. Today, if it is a 500 DFOs, you have to do the 500 DFOs. Time consuming, amount of circumstance. Human errors. Sometimes there's an artifact. Sir, you explore one and don't be there, I just skip it. Then only 5 fields are we are testing. Other field purpose for such setting. There also we are trying to do that. Then complex methodological procedures difficult to adopt by the stakeholders. Specific and sensitivity is relatively less. Sometimes say the virulent and non-virulent sir, or it's a virulent sir. These are all the problems are there. The vegetative life cycle, as I told, is very difficult to do that. So rapid and reliable testing is very much required on the system. Okay, we'll come to the real crux of the subject. Now, when you want to do that, either molecular diagnosis, normal PCR, there is a qPCR, LAMP, then ELISA, dot plot, LFA. These are all looks high-fi, great and learning service we can able to already we are back to at least young scientists are coming from the world and something that is done of but it's a very simple procedure. We not have much technique, very simple, which we can be able to do that. Now, how whether I'm going to talk only the theoretical, you can do the PCR, you have PCR. No, whatever we have done it in our laboratory, how we have done it, how we diagnose the developing diseases using the PCR, qPCR lab, ELISA. In the subsequent slides, I am going to show that. Now, we come to the PCR. If everything, when you theoretically tell me that, it will be very easy. The PCR not able to identify. When you take the gene of non-small 50 MD genome, the gene target ಮಾಡಿ how to amplify the PCR gene model. Whether you are amplifying the PCR, you should amplify only the Weber score. You should not amplify the host. Host also will have the genes. Weber score also. You should be very specific. You should amplify this gene should amplify only in the pathogen, target pathogen. So we have cross-checked that. This elongated factor 1 beta tubulin is a student of here at that time. She worked with Dr. Sindhil and Balavargan there, and she came there. She worked on that. We designed the primers. Then we tested with the Nosema bombasis, Nosema somasis. And when she was come here, Madam Dalita, she brought the sample. Thank you. And now she got the confirmation samples for that. We have brought the samples. We tested with all the samples. Whatever we designed these primers, we could be able to do that the PCR detection. So there is no artifact. If it is a positive, negative. Only thing is you have to bring the material, crush it, use phenol chloroform, purify, remove the genome DNA, pure the genome DNA, use all the techniques. This is a little cumbersome. I can do it in Kerala, I can do it in Dharmapura, I can do it in everything. She's all linked and Dharmapura chicken lab, we are going to use the PCR. You know, this is a really difficult task. Doing that one. Then, whatever you done, it should amplify only this Nosema species. It is not amplified upon its genome DNA. Other viruses, it's not amplified. It's a very specific whatever you have done. Now, second thing again, she going from some samples there from Muga. We did the multiplex, whether it's able to only the complex scoring which amplify or all other Nasima with everything there. And we have done it. We did the Subramani was working on that. So we made a complex multiplex PCR. Using the PCR, we could be able to Nasima infection with other infection also, like the CPV is a major problem that knowing that the CPV and the other virus pathogens are there, which we could be able to detect using the multiplex PCR. Now, there is a PCR is one another drawback. There is a test is called qualitative and quantitative. Qualitative, that is Nasima ide illa astray. And you come to quantitative, yes, Nasima is there. How much number of pathogens are there? It's very less or more, which we are to quantify that. So for that, we need further improved machine the real-time PCR. We were fortunate enough working Sericulture. We had two very good machines, which we purchased it. So we have quantified that. Why we need that? So very important. Mother mark. Yet we are testing. Yet whether it is having the prevalence score, it is there. How much quantity? In the first install, how much quantity? Second install, how much quantity? So we can be able to figure out how much of the quantity is there in the method. Is having how much quantity, how it is multiplying, the pathogen is multiplying, which could be able to do it during the qPCR, which we have done. Then another qPCR, further improved technique. When the COVID was there, I was scared. It's the same thing there. qPCR was already the proven technique for the identification of COVID. Everybody knows that. COVID bandhatanta, give you a blood sample, test it through qPCR. Then qPCR is a very difficult, it's a previous machine that time around 10-12 lakhs, getting the machines, but we can able to do that. So the Blackman assay, we designed the primer for our, whether we can be able to detect our method is there. Yes, we could be able to do it only 10 to the power there. When we use the qPCR, even 10 to the power 2 genome is there. Very least amount of reference score also, we can be able to do that. One more thing I'll tell you. As I told you, PCR which can be able to detect the vegetative stage. You cannot say it's a number of spores because spore is not a lecca here. We are going to the how much the pathogen is there. So we counted how much DNA is there. We call copy number. So even 10 to the power 2 copy number is there. So very less. 10 to the power 2 means 100 different spores are there in a sample. We could be able to do this detection qPCR. Now, very good. You got all the point now. You have developed the qPCR, PCR. Whether we can be able to transfer the technology to the field, a Tapatoli or either Hazara or your Mantipatta, can you able to transfer it? It's very difficult. Taking buying the machine, servicing, installation, keep the chemical in the -80, -20. Another is infrastructure you have out, huge expenditure. Now only put everything they have done with the biotechnology last 20-30 years, nothing has come to the facility testing. So I was a tremendous pressure. I was also, I was telling. I always feel that a managerial at least we think about the industry. We got the money from the government taxpayers' money. What we supposed to do that? Always I was a tremendous pressure. So I just from my heart I'm telling you. And you get the money, it's not like the get the salary, go and I'm responsible for that. You're very any meeting demoralized that say get biotechnology level. So I thought better I got to do something for this. Mardi. It's very easy. Solodal Yaritum, Eliavam, Ariavam, Soliavanam say you may be knowing that. It's very easy to tell. Let's go down is very difficult. Then lethal, okay, lab is the other man with the non that some of the hits are snatched, not snatched, that directly is snatched from us. These are given to CSR thing like so. So I took this one. Lab, which we are trying to do that. Lab is another simple technique. Very simple technique. You get the lab. Take few tubes. Already I made a ready-made mixture of enzymes, primer, 2 tubes. I have sent to the laboratory. I have sent to thermograph. Take the sample. Take the 2 drops in the tube. Keep it in the 60 degrees Celsius water bath. After 1.5 hours, there is a little color difference is there. You may know that it's a violet color, this is some blue color. If it's a blue color is there, it's a positive. It's a violet color is a negative. It's a very simple. But here also the problem is you have to keep, since it's a PST polymerase is there, you have to keep it in low temperature. You power steady, so the problem is that. So somehow I thought this is the easy technique which I can transfer to the field. Because whatever the technology is there, it should be useful to the industry. They have to adopt it. Then only our technology will be successful. So which we are trying to do that, which was taken by the University of Mysore. Then DNA, the cocktail, ready-made cocktail we are mixing. Keep it 1 hour, we'll observe the color. Based on the color, you can tell it is a positive or negative. Same color. Then we have optimized. We have to keep it 1 hour or we have to go to 30 minutes of later. So minimum, though it was sufficient for 30 minutes, on safety side we kept it for 30 minutes. Then we checked the DNA with the sample to confirm it. It's a conforming. Same lamp, left and right. But here it is highly sensitive. Anything, aerosol is light is settling on the negative control, they are also giving the positive result. They are struggle to troubleshoot it. Everything, though it is a negative, beginning is there. When you do keep on 2-3 times, when you do that, the aerosol form, the paper is formed that can initiate PST polymerase is fundamental to the genome median now, Nazima, everything is positive that. So we have struggled to do that. Somehow we come to the conclusion. Now, I the part. PCR I have done it, qPCR I have done it, lab I have done it. These are all the challenges I have. Last and because of a very simple technique which we can make it, you will make the pregnancy test. Lab to prostate it. I will do part. I show it. Take the urine, yeah, private female urine, suspected whether it's a pregnant or not. Put a drop of urine on the slide. Keep it for 30 minutes. You will get the 2 bags. It's a pregnant. Can we be able to do the same, really adopt the same technique in the government reduction? At the group of madam, she will test it. Sir, it is working, sir. I will give you 2 positive control, negative control. No positive control, 2 tubes, plus the strips. Then I will return the letter to the IRAC, DBT, to give the money because IRAC is that time I am thankful to Dr. Aslam, my call the person. He told me, please apply, you will be getting it. Then I identified one Bud Biotech laboratory. That Bud Biotech is famous for producing the it's a pregnancy test kit. So you have to identify the proper counterpart. It's not that. Scientific counterpart is a very weak counterpart. We cannot be able to get the nothing we will get. Then finally we will not get any. So fortunately, he was very successful establishing this one. There's an issue. There is a still he's producing biotech, but the other companies doing different thing. So he's a very famous for the diagnosis. So I requested, sir, it's not my priority. Somebody said, okay, it was specific for tomorrow. So I'm worried. So the later club we are I told him was this will be useful for. And finally he said, okay, bring it there. We can buy that project. It will be, we can take this simple test, grind the mark, put a drop on this sample holder. After 30 minutes will be there. Field level testing, user friendly, no recovered expensive equipment, very rapid, easy for the information. What we have done to show. See, this is this one. It is a board the method. Then it is so when you take it in the front test it. Anybody know what was the target protein? Anybody? I will tell you, it's a human gonadotropin protein, H6H3. When the female urine is there, 500, 600 protein numbers will be there. But how much investment they have done it? This pregnant woman have the protein called H6H3 more higher level. This normal, non-pregnant woman is not having that. They might screen large number of urine samples. What is the difference between pregnant woman and the normal woman? Finally, the pregnant woman should see the pregnant woman having the tighter of which she is more level. Better we can develop the antibody, we produce the diagnostic kit for the HCG. But I am the new man which I am trying to do that. Yes, sir has visited the South China University across Japan. It's a very famous place for the carcinoma. I asked him, sir, I am going to start with the diagnostic kit. Kindly give me which protein will be useful. ನಾನು ಇನ್ವೆಸ್ಟ್ ಮಾಡಿಕೊಂಡು ಆಮೇಲೆ if I am telling this is failure, again everybody will laugh say this is not working well. People, Cosmo is having more than 3,000 proteins are there. There is a score for proteins also, 20 to 30 score over proteins are there. You target score over protein 1, 2, 3. These are the major proteins we can be able to get it. Then we cloned, expressed, purified. This is all the other big contender with the help of Dr. Subramania, IBAB, could be able to do it. With Dr. Savitri also helped me in the use of science. So I am honoring those people there. So we can be able to do that. We injected, developed the antibody, because you need the two type of antibody. There is a polyclonal and one is monoclonal for the diagnosis, which I am not going to explain yet. Just if I talk about only other principle, it will take another 45 minutes to talk about the principle. So here, the simple technique. This is a Bebride score antigen, primary antibody, first antibody, secondary antibody towards this one, you will get the color. If the Bebride scores are there, HRP color will be there. The Bebride scores are not there, so you will not get any color reaction. Simple ELISA reaction, which we have done. Then we have tested should not be non-specific. Bebride color sir, it's all there, sir, it's not working there. Everywhere it's color is coming, color is faint, color is strong color. All big problems are there. So we tested score over protein 1 and 2 are working there. BSA and non-target is not working. So we thought it would be the good target protein. Our antibody also working. We are confirming. The simple technique. Take the CRC, put the engaged will form. Why I am telling you, mother mark will be the known scores will be there. When you take the CRC, the score may be present or may not be present in the young larvae. You take it, grind it, take the suspension, put it on the slide, you will get the color reaction, yes or no. So we will try to do that. This is not theoretical. It's we have done it by night and day, which I sit there. How to do that. We are not working there. Some chair of some chair, they're very casual. They some about night and day, which we are trying to force them. We have seen that. So we had able to do that. We had developed Andrea Mylita, Bombix Modi, all which were working there for the diagnostics. So now, sir has given out the 2 lakhs for the belief. And the field trial, probably shortly we will be going to get it for the diagnostic of this one. Then in parallel, yesterday was telling, VC was telling, publication is the yardstick for the judge your the caliber, merit caliber. You can publish a paper in the pediatric journals or other journals, and always you should be very good. Spring well, Elsevier, top class journals, very good. And my publications will be always will be the good journals of that. Spring and publication. In addition to the president. But thank you once again, sir. Yes, call me. Sir, BD sir, I do say, sir, Harling, sir. Planning a portal, university, they are calling, sir, call me. As I want to sit there, very good, sir. Thank you. Thank you. I think there will be no answer the questions now. Not it will be there. Cannot tell that. All questions you cleared.
Anyway, very good presentation. How conventional to PCR, multi-PCR, and your genome-based and protein-based diagnostic. Really, it was beautiful.
I have one question. When can we get that?
Madam,
It is very useful for all seed production centers.
Sir, in the
Only I wondered, anyway, that
I will send you, sir.
but why the Alexa could not perform? What was the reason behind it?
Sir, Alexa, there's a electrophoresis of another version of Alexa.
Yes.
Okay, Alexa, you get the antibody for antibody, second antibody, conjugate the whole Alexa later grades way. All the things that we have to install in the The main purpose of this project is it should be useful for the portfolio. There is no reason, but I will tell you, sir, in order to validate the counter checking, we use the Alexa, sir, left and right. We use the Alexa. It's working really well with Alexa. Whether we could be able to reproduce in the lab possible. Alexa.
Less number also.
Less number, PCR not the best, madam. Today I am telling you.
But PCR also funded it.
Madam, now the technology is becoming very simple. The working for also very ready-made samples. These are the best method. Now we are going to get it the first surveys are called you please develop establish separate molecular parameter, small parameter. And if you try to get the once if I show them, it's a very easy. These young people are very eager to work on that. So I hope they will try to do that. Come on, now about this one, the permission of the director, I put out 500 kids, I will be sending. At least 500, 300 kids.
Never to test it.
Yes,
Discuss that.
I will be sending it.
What's the cost?
Madam, this is as on today, then the fancy bill is around 50 rupees, madam, about 50 to 70 rupees per. Different company, different design. Teams are also 50 to 60. It's 60. Now, it's over, maybe maximum 50 to 60, that's all. More than that, not very long. So very simple, cost effective. It's commercial, already it's released, man. The customer is behind us. It's all with the now direct in charge of this. We are probably he will. Sir, also.
We want to have actually, they are telling us we want to have the sample across India. One person wanted to have one control message. They want to commercialize, but you cannot take back. You want that cost is to make it. One person test will take out.
Some of the mark can be only
Any other technique came other than protein based now? Any advanced techniques?
Advanced technique, which as I told you, sir, I'm going there. There are some techniques out there, sir. Like CH believe that diabetic medicine has spoiled my memory power. Really, I will not be able to remember it. It's a very simple, sir. The wheat based, like a wheat based, sir. They want to October, sir. October,
After you were having one concept.
sir, when Sir was there in Barampur, he proposed that one after. But that group is very strong, sir. It's a micro-protein control laboratory. That's they developed the top of the best without pan advance. Pan and TNT. So,
Can we test this one for other diseases also, or what it would have been?
good question, sir. Good question. Good question. It was mine, this one. When we talk about Robert's Mori, we have introduced a major disease, all flu. Then, your maximum extent that is that one deviated basically, which is that now
That's correct.
these three are we want to combine. At least for the BMLPB and this one, we want to combine.
It's quite common.
Yes, ma'am. Yes, ma'am. Yes, ma'am. Especially when you go to the CRC, you can put the lung lymph compound. If it's a positive part, then you can you can discard a lot of that. We start with this. The industrial farmers are very curious, madam. You are not seeing it.
Yes.
If any problem is there, they are right to number the crop loss because they are counting the amount of remuneration and money. So it will be very. So we will tell them please discard the lot at the earliest. Get the 3 not then be fine. So we are going to make a combined diagnostic. Probably my juniors, so probably they will be more very, very smart, very efficient teams for the sector. I hope they will try to. Thank you. Thank you.
Thank you. Next person is Ravish Kumar. So the sustainable approach for pest management is informed and West Bengal. Ravish, please, 5 minutes, 2 minutes for this discussion, presentation.
Yeah. Last week syndrome to that's not because it was not another. I was everybody doing something. I used to think that's
Touch it out, touch it out.
That's what this is.
Start a picture. Start out.
no problem. One friend of
That's all, let's let you know very important.
Start out. Start out.
Permission of all. I'm sorry to say this because I requested for closed here, but somehow I got the opportunity to present for the presentation. So, actually, my work is on polyisobutyl an approach for pest management in silkworm rearing in West Bengal. So actually, West Bengal is 50 years back sericulture compared to South India, because they are practicing very much traditionally. They are doing the sericulture because they don't have a proper rearing house and all. They are doing with their dwelling house that to thatched roof and mud wall and very. I can say they are very economically cheap. This is the main primary things to design this one. And again, if it is becoming very traditionally built, so they have a lot of problems with pest management also, like ants, lizards, and even some rat, because their house is so, you can see that slide first one, first photo, that the house are very economically very big. And when it comes to mulberry plantation also, there is no spacing. Now they are initiated to look for 2 by 2 spacing and all. Most of the farmers are doing close spacing, like 50 years before farmers are doing like that. Although these things were there, the limitation was there. The farmers are not that much interested to monitor the pest and all, and they are somewhat neglected to look after all these things because they are earning for additional 10 to 15,000 rupees per each crop. So we thought whether this is some agriculture company like Barracks Magic glue, they have developed a glue in such a way that to attract the pest in agriculture crops. So we took that the same glue and we utilized for both silkworm rearing and mulberry pest. Because for controlling mulberry disease, again we need to tell them to take some chemicals and all. And again for silkworm rearing, again we have to tell some of the things to adopt everything. So we thought whether if we apply properly this glue, it can be a solution for both the things so that their economic danger will be reduced. So what we do, we selected an ideal mulberry field similar to that, and we purchased a chemical based company having polyisopropyl chemical formula, and it is available in the market in the name of Barracks Magic Glue. So what we prepared, we applied in the yellow card, it acts as a sticky trap. Similarly, what we have done, they will constantly change the Chandrake, they will keep the mountage outside it, and the rearing stand is also somewhat permanent things. So we applied that in the base of the things and we observed the things. So on observing, we identify almost all the pests of silkworm that get attracted to that group. And when we kept in mulberry field, many of the sucking pests which are dangerous, usually that leaf-eating caterpillars are not much effective there. Usually sucking pests are more as with little bit of my observation. And many of the sucking pests were caught in that. So we thought, and this glue have a specialty that it would resist for high temperature and high humidity, even in the rain also, because the West Bengal and Assam season we will have very high temperature, and it is waterproof. Also. When it goes for continuous rain also, this glue has kept their sticky nature as such over the concentration. And we observed many of the pests has been attracted. Still, we need to improve the standardize the things, how it effectively improve, like keeping the loose and all to along with this trap. So this is the work. We found many of the pests and diseases that was attracted each other. By thinking of this in future also we can see like Tatan also in the forest based things. So we can't apply all the things. We can put this glue around the tree trunk so that it can avoid the ants and termites and many more. And this is an eco-friendly approach. While doing the crop also it will sustain and it will sustain till 4 months. As one observation we have done in south only with our farmer. So it will sustain till 4 months. By this, we can conclude that this is an eco-friendly approach and it can really help the farmers in an economic way. I can't say this is 100% manageable, but somehow it will manage the pests and diseases, especially in the West Bengal. It is around 200-300 rupees. So we can tell them it acts both multi-purpose, I can say, for both silkworm rearing and mulberry. This is the initial work we have done. Still, it requires a standardization. So I thank all of you for giving me the opportunity to present this work.
How many seasons you did it? Like,
Sir, only one season.
one season.
One of the.
Which is major season for many first? No.
Yes, sir. Actually, this is the thing we observed. Only one we see. Only one farmer we observed.
What are the question which we got in this?
Yes, sir. Ma'am, I have just white flies for incidents were there. That we got
During that season, what was the just because all the season, all the best.
no, that could be I need to be honest. I just selected these things. We go for literature. We found that many pesticide.
White flies only move around the acids also.
Yes.
But other pests and mealybugs, they are inherently in that in persons.
Yes, ma'am. Actually, that farmer is not maintaining much well. It's just an open field. Along.
White fly is specific to Sericulture.
Yes.
It's not white fly. Other insects they don't have, they have it was.
Yes.
It suits that's you are the.
Actually, 4 months we took none.
You cannot.
After 4 months, we took an observation.
Maybe, but other insects they will be. Yellow stickers.
Ma'am, all sucking pest I observed. Actually, I took with some new scientists. I am not basically an entomologist. So
No, actually, mealybugs don't move from that.
no, male mealybug they told.
Male mealybugs move, but most of the, I think it is effective only for white rice. The same one we did is the plant of mud. And we had a TOD also.
Well, actually, I want to say that it acts both for silkworm rearing and the mulberry.
Okay.
I'm not stressing for only the mulberry field. Once he purchase these things, he can use with any yellow sheets. So that will be economic. My idea was we should be economic to them.
Since it is yellow color, pussy
Yeah.
fly
Yes, sir. Yes, ma'am.
you have not mentioned, yeah, attracts rats.
No, ma'am, we are right.
Okay.
But they are not like us, ma'am. It is completely very. You see that.
Good attempt, the rearing and mulberry field combined. But this is here because white fly is common, possible, yes, it also. And some extent, you are.
Servicing them.
Is Amnath use the rain of the use for visible use.
So many rearing issues are given restrictions.
Actually, this is not an experiment. Only we purchased that glue and we kept it.
Must experiment here.
So it is initiated. It is an initial idea. Need standardization. All by keeping everything.
It will attract and it will die or just honestly attract. It will stay there. By force, it can escape.
The gun is so small.
But it is because it die later.
Answer.
Yes.
When it stick over the long duration, it is the immediate state.
It will force to escape immediately. That you are improvement. No, it will die later.
Even the rat, if it is because it can't escape. That much strong it.
Like rat, no.
Yes, sir, yes. Sir. That is also blue only.
Golda, tomato, like that, sure. Rat or fish will be there overnight, tomorrow morning you can see. Okay, good approach, but you have to try so many generations because the insects seasonal wise varies. Because they like sporadic, some of them are in the winter season, some of them are in the days. Like that it is there. So you have to find out which one is more effective to minimize the level of insect. And then look for the next generation, what would be the best alternate rather than isogluten group that you have proposed. That way, that helps. But you have to give recommendation to the farmer at the end. You just try and keep it everything in the lab, and when people are not aware of it, farmer is unable to practice it, it becomes no use. It requires you have to try so many trial and error before insisting upon the results. Okay.
Next person is Dr. Sahana, scientist B. Topic is the effect of sulfur on fecundity of silkworm hybrid. Let's see.
Yeah, we should move the slide. Slide more. This is 20 PM. Here it's I can change the height. You can make the distance based on your convenience. You can make you can keyboard is there. Huh? Okay. Chill. Good morning, everyone. Respected chair, co-chair, and senior scientists and your friends. Myself, Dr. Sahana, working as a scientist B at P2 Basic Seed from Dharmapura. I'm being in an NSSO, that is National Silkworm Seed Organization, which is majorly contributing for a silkworm seed and supplying to farmers. Actually, for mulberry production and for the control of mites in the field, we are recommending, scientists are recommending that vetiver sulfur for the control of mites. But what our seed producing scientists observe that after using the sulfur for controlling a mite, if we produce the silkworm by using those mulberry leaves, there is a lesser production of seeds in the drainages. So this observation is made from a few of our scientists. So just to confirm that we had a basic study here. By using the same chemical that is a vegetable sulfur, for that we have studied that what is the effect of sulfur on peak content. Since so the objective of the study is to know the effect of sulfur on the picability of the silkworm. Coming to the methodology, we have used B1 mulberry garden for the spray. That is after 45th day of this pruning, we have sprayed the vegetable sulfur. Here for our study, we have used FC2 silkworm hybrid since it is one of the parent for the double hybrid. And vegetable sulfur, 80% WDG is being used, was used for the study. And for the treatment, we had a 4 treatment, whereas one is a control with the water spray. T1, that is treatment 1 is 1.5 gram per liter of water. T2 is a 3 gram per liter of water and T3 is a 6 gram per liter of water. Here the treatment 2 is a 3 gram per liter of water, which is being recommended for the control of mites in a patient. So with this treatment, what we could observe that after till rearing and up to cocoon production till moth emergence, there was no difference between the treatments. During the whole portion, that is when we observe the fecundity among the treatments, control it was around 532 number per laying. This is the average data I am presenting here. T1 it is 527 number per laying, whereas T2 which had a 3 gram per liter of vegetable sulfur, which is being used for the control of mites, has had given 363 number per day. Whereas the T3 which had a more concentration of vegetable sulfur, again there is a change in trend in case of both position, that is a fecundity, it is around 471. If we look into the reduction in percentage of fecundity, we can observe that at the T2 concentration around 31% reduction is observed. So from this it is clear that the recommendation which we are recommending for the farmers with the control of mites is affecting seed production and drainages. Farmers are putting same effort from the day of brushing till cocoon harvesting and also from the drainages till starting from cocoon adjustment till the moth emergence, they will be putting same efforts for the same quantity of cocoons which are received from the farmers who are used both the sulfur and without the sulfur. Was after moth emergence, if they won't receive the expected quantity of seeds, it will be lost to the drainage. So it's clear that the recommending the dose which is being used by the farmers for the control of mites is affecting the silkworm seed production. The same treatment observation here we can see that the first photograph is indicating the control, but this is the T1. In the T2, that is 3 gram per liter. We can see the poor layers compared to other treatments, and also even egg-laying pattern is sparse compared to other treatments. So with this, our study, it is clear that 3 gram per liter of concentration is being affecting the silkworm, that is fecundity of the silkworm. And for the silkworm seed raider, it's better not to be vegetable sulfur at a concentration of 3 gram per liter, or else we have to find out the concentration which can be controlled, which can control both mites in the pit, and it should not affect the seed production in the drainages. So this is the outcome, and also we can suspect that the same concentration may be affecting the oogenesis process in the Pomix mori moth, or the same concentration might be affecting old function even after oocyte formation. These two are the things where we can study in future. This actually for FC2 we have studied, even we can study for the parental basis in future. So I thank our team and NSSO for their support, and also I thank the organizers for giving us this opportunity. Thank you all. Yes, ma'am.
Presentation, but you go back to treatment.
This one, woman.
Treatment detail. 6 grams, why it is not affecting?
But that's what, ma'am. Out of curiosity only, I kept more concentration.
6 grams, you are showing 471.
Yes, ma'am.
But 3 gram, 320 in between, no?
In between 4.5 you were about to give, that will be studied in future, ma'am. That I have to study.
Why it is not? Because control and T1 is somewhat on par.
Yes, ma'am.
And 3 grams less
Less
fecundity.
But 6 grams, if you are increasing the dose, it is again frequency is also.
Increasing, ma'am, that needs to be studied, ma'am, because after observing this data, you have to articulate why it is increasing the pattern. That's why I said, we have.
Whether we can recommend people.
That's what, ma'am, I said, whether we have to check one more time, like standardization need to be done. Either it should not affect the fecundity in Bombay's more, or meanwhile, it should equally take care of the biopsy. Only one trial I
You
did.
have to repeat that. Right?
Yes, we will do, ma'am, that's what we do.
Because this thing.
But along with FC2, I have used other silicone rays, so I will also FC1 and CS.
You have to repeat some ratios and repeat it. If it's.
We had a more
find
replication.
whether it is, this trend is repeated.
Yes, ma'am, that we have.
The replications is going on. How many?
4 replications I have used, ma'am.
Actually, we cannot recommend T1. Supplement.
One concentration between T2 and T3,
Yes, sir.
then you prepare T5.
Okay.
Make it 4.5%.
4.5.
Otherwise, 7.5% after T3, then it become T4.
Yes,
So
sir.
try. And because there is a gap of 3 to 6
Yes, sir.
and 1 to 3, then so it might be one where you can optimize the exact concentration. That will help for the fecundity rate, and also the
Whites
whites population we can reduce that out to 5. Gas is more. And sulfur is actually toxic, that may affect the fecundity rate. What about testing?
Hatching wise, no problems.
You have not done.
Actually, we have not done in my experience, sir, in my experiment. But in SSPC, we have observed that there is no hatching difference with the sulfur usage batch because of the batch. That we have observed, sir. There is no hatching difference.
No. What would be the rate of hatching percentage?
It is 92%. As that of control only, we are getting hatching in case of
That's
sulfur.
also useful because both of them are parameters.
He said that is just
You could
the
see one
ancient those
fecundity
things.
not actually that is not correct. When you look for H parameter, you see H parameter, double parameter, larval parameter, like that you should not just concentrate because it is also immediately it is a consequent stage is hatching.
Yes, sir. Yes.
That you have to look into that. That may be disturb the fecundity also, even it is disturb the fecundity. Even definitely there is a proportionate effect on the
Hatching.
hatching of the egg that you have to see. Okay?
Just for suggestions, since you are going to confirm it that
Ah.
you can, why not you can try different sources of sulfur? Instead of trying only vegetable sulfur, you have lot of other sources. Even this quantity required will be very less when you try with some other sources like elemental sulfur. Even single superphosphate is a sulfur source we can take, and gypsum is also sulfur source. They are very cheap. Better the sulfur is very cheaper, maybe 70.
Yes, ma'am, 70.
Gypsum I have 6 rupees per kg. So the solubility wise both
Their relevance is
Mites.
we are using sulfur, we are using for controlling of mites. In that context, she has taken metals.
Just since it is going to be a research, they can try. They can reduce it also. Quantity you can reduce it, economics you can reduce it.
They have recommended upon in terms of pest management. They have consumer, they cannot see. If they are working seed bottom,
Yes.
they get the low number of fecundity. They are not a small set taken. Anyway, that Mala taken on comprehensive plan. Like,
Actually, for the date of spraying, the crushing date is 5 days for all the treatment. There is no difference. The recommendation is of spraying.
only
No, the leaves, spray leaves. Yes, sir. From the date of crushing to the spraying.
For that,
Sir, all the treatments are imposed on the date of.
We wanted to know what is
Same day, yes, sir.
What's your waiting period?
5 days, ma'am. That is only recommended for white control also, so I have used the same waiting period.
The number of sprays, how many number of sprays you have to?
One spray. One spray 5 days before brushing.
Anyway, this is a preliminary work, the attempt test. Actually, we wanted to know whether impacts are there. Impact is there, it may some minor differences. Thank you.
Next presenter is Dr. Ranjit Kumar, scientist B. Topic is evaluation of botanicals on DM NPV infected mulberries in form of fungus.
Good morning, everyone. I'm going to present a small topic of my PhD thesis. That is evaluation of botanicals and PMLG infected mulberry silkworm bobbing story. We all know that mulberry silkworm bobbing story is a susceptible to insect disease. It can be easily affected by diseases. Among diseases, virus disease accounts for about 70% total losses in the industry. So we are focusing on controlling the disease using botanicals. Why we are focusing on botanicals? Due to usage of chemicals as disinfectant, it is biodegradable and carcinogenic. That's why we are focusing on management through botanicals. The biomolecules present in the botanicals such as phenols, flavonoids, they may possess antimicrobial activity and may enhance the resistance to the silkworms against the diseases. The objective of research, I have taken 3 medicinal drugs and effective generally. Eureka missionaries, Kilanelli, and Sorale Family. The earlier studies in Sorale Family, they have taken seeds for controlling the disease. But in this study, I have taken Sorale Family leaves and I have extracted the methodology. I have extracted using a chloroform. Today, I have taken 3 acetone, methanol, and chloroform. Comparing those 3 solvents, chloroform uses some more biomolecules extraction. This I have taken 3 sampling virus extraction of 10 to 1,000. They are given as first field of third instance silkworm larvae. Botanicals are application by studies. Grassary affects in all instas, but it is more common in fourth and fifth instar. So they are given viral suspension on third instar first day, and that the botanicals they have given on second day of third instar and first day of twice. The worm spread with the BMNPV alone as well as treated control and untreated control supplementation. From the I have taken 2 different concentration of 3 botanicals, 500 and 1000 ppm. From the table, it is clearly on the Sorale Family of 1000 ppm concentration has shown very less larval mortality compared to treated control, BMNPV control. And followed by 500 ppm of Sorale Family. And the Philanthus delivery Eureka is also given some positive relation towards the control of this. But Sorale Family stand first. And the larval weight also we can see it is on par with the untreated control. 4.8. I'm seeing that the treated control it is 1.98. More or less the worms are completely died at the peak percent. And some are went for non-splitting. This is Kapur rate share ratio. You can see that it is after the untreated control and Sorale Family for 1000 ppm on time. And when I study the what are the antimicrobial or anti-inflammatory components present in the Sorale Family for this, I have taken for GCMS, CCMP, and some more than 30 plus compounds are identified. These are the major which expresses anti-inflammatory, antimicrobial, but thing I don't find the most antiviral components. If it is been like we could have more next it has more anti-inflammatory, anti-inflammatory. So this may enhance the control of the disease. And in this, you can find the antimicrobial and antiviral properties of the botanicals through different modes of which can be applied. So what we can Sorale Family things can be applied for treatment of grassy disease. It was for that chloroform acetone, methanol, and chloroform 3 I have taken. And comparing this 3 chloroform extraction is given more compounds extraction through the extraction. What is the name of the breed that you have chosen? Silkworm breed, silkworm double. What is that name? You have mentioned there. I have mentioned. Because the couple weight is very, very low. 1.2. 0.65. Those are virus induced. That you have to indicate. I think that is index, evaluation index. Right? That is evaluation index. That is not represented in terms of graphs. You represent it in terms of graphs. You should not indicate evaluation. If the evaluation index is different. Say graphs, it should be actual weight, partial weight, shell ratio also. Ratio is very less. Like ratio convention. Because the parameters are ratio differences are multi-voltage, bi-voltage, cross weights, double end weights, many. You have to mention the breed also. 1.65, which is the name of the breed? Krishnavaja. Approve the sir.
No, I will explain first. Normal, we have 2.43. Those are very good. Normal. When we treated with in people, it has come from 1.65. The untreated control source for it. Next column. 2.43. The Chennai thing is when we treated with in people, so though it's supposed to be 1.21. Due to empirical observations, it was increased to 1.66. First to because it already 2.24. That is steep point. Okay. 2.1. You may be taking the press card. The R next to the so the data checkment. ಈಗ ನಿಮ್ದು ಚೆನ್ನಾಗಿದೆ ಅಂತ ತೋರಿಸ್ತಾ ಎಲ್ಲಿ ವರ್ಕಿಂಗ್ ಮೆಕ್ಯಾನಿಕಲ್ಸ್ ಅಲ್ಲಿ ಎಲ್ಲ ಕಡಿಮೆ ಇದೆಯಲ್ವಾ ಚೆನ್ನಾಗಿದೆ ಅಲ್ವಾ ಗುಡ್ ಮ್ಯಾಮ್ ಮತ್ತೆ ನೆಟ್ ಇಂಡಿಕೇಶನ್ ಅಲ್ಲಿ ಏನಾದ್ರು ಅಂತ 2.43 2.24 T5, what is the indication? 2.24. And T8 is 2.43. What is the indication?
That is control.
No, virus is given for this 100 control. ಈಗ ಅದು ಕಲರ್ ಅಂತ ತೋರಿಸಿದ್ರೆ ಅದು ತುಂಬಾ ಜಾಸ್ತಿ ಏನು ಅರ್ಥದಲ್ಲಿ ತೋರಿಸ್ಬೇಕಲ್ವಾ ಇವನ್ ಲೆಸ್ ದ್ಯಾನ್ ದಿ ಕಂಟ್ರೋಲ್ ಆಲ್ಸೋ ಇನ್ ಆಲ್ ಪ್ಯಾರಾಮೀಟರ್ಸ್ you have to mention this T8 as in written. That is in white and black. You, according to any findings that you are any possible actually part of sir after this three parameters I have taken thoroughly I have blended with the titanium and the aromaticals. You try the lesser thinking. Go for thinking.
They're asking.
Okay,
Next. Microbial. There is no antiviral application.
in this only for antivirus. Based on the earlier reports also, Sorle cornifolia leaf doesn't have more paper. Leaves of Sorle cornifolia has more. For peppers. Many research are in Mysore river. But I have taken leaf, so you could see what is there in leaf. What is good? Because you get lesser concentration. Instead of 500,000.
Maybe microbial. Maybe virus or bacteria also.
One way or the other way, in one character. All the characters negative. Based on studies, I have fixed it 500. Control is the best. Don't do experiment then next time. No one said this is to come to graph.
Okay.
Very good.
Next, I invite Landa Preeti to present a paper on the related information.
I have used three solvents, sir. Acetone, ethanol, fluoroform. Are those three solvents? Fluoroform extract has given best result. Result on extracting more biomolecules. No.
We tried it. Under program. TLC. TLC, I'm saying the TLC layer, I have centered more biomolecules in chloroform. But more articles are there on acetone. Some are telling acetone will give more extra. But in my study, chloroform was given many TLC. But chloroform is given more.
Chloroform final method is not alkali salmon. It's quite sensitive method to extract majority of the powder and botanicals. So you can purify used for HPLC, TLC, etc. Good technology.
Now I invite Lata Priti to present paper on regulatory personnel of the class of disease adolescents.
Working as scientist in Central Sericulture and Research and Training Institute, Mysuru. They are here to present on predatory potential of Lactobacillus palaceens on broad meat. Let me begin with a brief introduction. The pest Polypharmacotoxin sandis, which is more commonly known as the broad mite, has gained the status of a major pest and still it is persisting as a burning sensation, as it is known to affect the succulent tender leaves, making it unavailable for the consumption of silkworms. As it is a known fact that usage of relying only on the pesticides or insecticides to address these pests is highly unlikely when it comes to mulching. So, in such a case, it is the more appropriate alternative would be the employment of biological control agents. Lactobacillus palaceens is one such biological control agent. Though it is being proven as a promising biological control agent against many other pests, when it comes to our pest of interest, the broad mite, it's still being unexplored with its predatory potential. So, our study was aimed to evaluate the predatory potential of Lactobacillus palaceens against the broad mite. So, for the study, we took the laboratory cultures of Lactobacillus palaceens, which is maintained in the pest management laboratory of CSRDA Mysuru. And throughout the study period, the temperature and the relative humidity of the lab was monitored. The study was initiated by obtaining the eggs of synchronous oviposition and randomly selected the newly hatched eggs, that is the day-old nymphs we have taken for the study. The study was designed in such a way that two different assays will be carried simultaneously. One to evaluate the predatory potential against the egg stage of the pest, and the other assay to evaluate the predatory potential against the active stages of the pest. And for each assay, 5 replications were left in order to ensure the statistical reliability. So, from the by visually interpreting the observed results, we found that throughout the instars there were a change in mean. So, in order to quantify the relationship between the predatory capacity between the stages, 2 different stages, that is egg stage and the active stage, we studied it and we found that for both the stages there was a positive correlation, but we also came to know that there was more correlation between the predatory capacity of the predator and the active stages of the pest. So further, in detail to study the interaction between that, we plotted the interaction plot and we came to know that was a clear shift from the preference of the predator, which was like from the, it showed a preference to feed on eggs in the early stages of instar, and as its growth advanced, it shifted towards the active stage. So further to confirm it statistically, we conducted the ANOVA test, and in this, firstly we kept the factor 1 as the different instar stages. 5 different instars are there. So considering that as a factor 1, we couldn't appreciate any significant difference in the predatory potential. Then coming to the factor 2, that is the stages of the pest. One is egg stage and the active stage. When that came into the picture, we found that there was a significant difference in the predatory potential of the predator, that is the bee parasites. So we further studied the interaction of different instar and the different stage of pest. In that, we significantly found that the early instar, that is the first instar, preferred more of eggs, and as it advanced in its growth,
there was a decline in the preference towards the X stage of the pest, whereas which was vice versa in case of active stages. The advanced stage of the instar showed more preference towards the active stages of the pest. So finally, from the study, we could conclude that the bee parasites would be a promising biological control agent against the mites also, that is the Polyphagotarsonimus latus also. And also by observing the stage-specific preference of the bee parasites, we could hypothesis that by releasing, it would be more appropriate strategy to release both the mix of early and late instar to target both the egg as well as the active stage of the pest. So that this could enhance the predatory efficiency, thereby effectively we can control the pest. Thank you.
Open for discussion.
Yeah, it's already been practiced in the Namaste. For thrips, they have done one, and for mites also they are recommending, but this is a company like that also, ma'am. But the study was made to find out which it is targeting, because when we are relying on the biological control agent, so we can't completely eradicate the pest. If we target the pest, like both eggs and adult, if we target and we give means, it will be more effective than blindly giving one stage of the biological control agent. If we give a admixture of both early and late means, early will prefer the eggs, so that it will function as ovicide, and adulticide we can give the late advanced stages of instar, so that at that time, at the moment only, we can that control will be more effective.
So, if this is for my knowledge, what about the mites occurrence, seasonal occurrence? Which season mites will remove?
Now, with the increase in temperature, it is getting more, but it's prevalent all throughout the year. But when the summer season or the temperature is so high,
if you release, so only during the high season times, the predator will be safe in the environment. What is the other measure you will put into the system for maintaining the residual population of the biological control species in the environment? What is your suggestion?
well, actually predator means it can have different hosts.
Yeah.
So if like there has been one study that it has that other preference also. If other most preferred host is there, means it will fly and go, they have the wings also.
Alternate host, what are the alternate host you have to study and keep it in the system, so that the residual population of the biological control organism which you have released should sustain in the environment. That is very important.
Good. Where you are working your task?
Yeah.
Myself says that in customer management.
Basically, veterinary doctor, sir.
Oh, that. DBS, yeah.
Next presenter is Dr. Priyadarshini. Topic is evaluation of wound healing property of sericin in rats.
Good afternoon, honorable present here. Myself, Dr. Priyadarshini, working as a professor in the Department of Sericulture, Forest College and Research Institute, Medipalaya. I hereby to present my research findings of the topic on evaluation of wound healing property of sericin in rats. All we know that the sericin is produced by the silk form. It's a natural biopolymer. It totally accounts for 20 to 30% of the cocoon weight. We know that this sericin is rich in biological properties such as biocompatibility, biodegradability, bio-antioxidant activity, anti-inflammatory activity, antimicrobial activity, and wound healing properties. Though it is rich in biological properties, we are not at all using, we are not recovering from the real water. So with this view, the current study is focused with the objectives on to extract the sericin from cut cocoons as well as sericin water, and to find out the evaluation of wound healing activity of sericin in diabetic induced rats. For this work, I have extracted a sericin with the using of cut cocoons at high temperature 120 degrees Celsius for 60 minutes. After that, the solution was allowed to cool filtered, followed by certification, then under spray drying for to get fine powder of sericin. Then next one is evaluation of wound healing activity. For this, actually, this work was carried at PhD College of Pharmacy, and this work was approved by the Institutional Animal Ethics Committee also. Here we have taken open excision type of wound. For this 400 millimeter square wound was created. Then 4 treatments, sericin 4%, sericin 8%, and standard neosporin and control without treatment we have applied. After application, the percent of wound area on first day, 4th day, 7th day, and 14th day was measured. Coming to the results. Actually, among the 4 treatments, the sericin treated rats showed improved wound size reduction than standard neosporin and control. Actually, this is the first day, then 4th day, 7th day, then 14th day. You can see the 100% wound area, but during the 14th day, in case of 8%, we can see the 100% wound reduction because this sericin helps in preventing the prolonged inflammation of chronic ants, as well as this sericin absorbs the excretion secreted by the wounds. Actually, wound healing process occurs in 4 stages. One is hemostasis, followed by inflammation. Third one is proliferation and remodeling. First stage in case of hemostasis to prevent the blood loss. Followed by inflammation, here cytokines are produced. Then proliferation, here fibroblast growth factor and fibroblast growth factor and collagen production takes place. Finally, the development of scar will occur. Yeah, we can see this is the first day. We can see the wounds, and on the day 4, here we can see the wound area reduction in sericin treated groups 4% and 8%. Then 14th day, we can see the difference between control and the sericin 8%. We can see only the development of scar. So that is the last stage. That is remodeling. So this sericin helps in collagen production as well as fibroblast production. Then re-epithelialization, then as promotes the proliferation. So, based on this study, it is proved that the sericin could be used as a natural wound healing agent instead of going of synthetic drugs. Then, apart from this healing bottles simply discarded by the healing industries. Instead of this, we can recover the sericin and can be used for not only for cocoon production is not only for textile industry, but also for diversification of non-textile applications such as the recovery and use of sericin not only reduce the environmental impact, but also it enhances the great potential value in diversified industries such as pharmaceuticals, food industries, and cosmetic industries. Thank you.
For discussion, any questions?
Previously, whether sericin alone or in the conjugate bit.
Yes, sir. Sericin plus fats, these fats. These fats.
Then 8% will for sericin.
Yes, sir. These fats actually, sir, generally they used to include coconut oil for moisturizing, but sericin is a factor as a moisturizing agent, so we are not including coconut oil.
Yes, coconut oil, you use this.
Yes.
But we should cost a bit because coconut oil is.
But we are not included coconut oil, only fats.
No, that is what replacement of coconut oil. Who are the funding agency this one? In university or?
DSC, sir.
DSC. Oh, DSC self-public. Okay. This what are the other regulatory process? One is animal ethical committee.
Yes, sir.
Then other than that.
Sir, now we are speaking to the College of Pharmacy, sir, to go further, for another formulations. Actually, we formulated only ointment based.
Okay.
So now we are going for hydrogel, clean based.
My question is, sir, what are the regulatory process?
Ethical
is common. Ethical is only for institutions approval. Other pharma, I mean, making the pharmaceutical application. What are the pharmaceutical?
Actually, we collaborate with that pharmacy college. Those process will be taken by the institutes.
You have idea, no? You don't know because there are cytotoxic will be there, so many will be there, genetic this certain mask. Oh. Very good. Flooding is over now.
Hey,
Now second
now
phase,
we are doing, sir.
second phase you have undertaken. Okay.
Thank you, sir.
Why is Sericin taken? Why can't pipeline?
The background is heavy change, but
Okay.
there is an extraction is very easy compared to that.
Yes, sir. Really, in more of your advantage is in pipeline.
Because sir, there is a, you can extract very easily because started with the reading system. That is the main point, sir.
Heavy chain molecules are there in pipeline.
Yes, sir.
There is a low chain molecules are there, molecular weight is there.
Another complex protein, sir. We have
Actually, hydrophobic, there is hydrophobic, sir.
Ah. It is a core protein that is very different.
even they are called pipeline also both hydrophobic and hydrophilic. Some chains are hydrophilic. Like chain is that
Yes, sir.
it is hydrophilic.
And now, Dr. Likith Gowda will be presenting on right, a Bombex Mori infection model for evaluation of antibiotics against human dental caries after the streptococcus mutans. Please.
Welcome everyone. Today I'd like to present a part of my PhD thesis work. So generally, all of you know that the silkworm is economically beneficial insect. It gives the jobs to around, it provides the jobs to around 90 lakh people. So among that, silkworm gained the importance in the medical field aspect. So in that line, most of the nowadays many scientists are trying to use silkworm as a model animal for various diseases, for various development of a vaccine, for the development of a antibacterial protein. So like that, the silkworms are being using in the medical field also. So in that line, we try to use silkworm as a model animal for a human dental caries pathogen as Streptococcus mutans. Streptococcus mutans is generally a gram-positive facultative bacteria. So it's a primary habitat for the fungus. So it majorly causes a dental caries. So it's galvanizes the tooth surface and it damages the hard structure of the teeth. So among that, other than that, these Streptococcus mutants have the capacity to invade the circulatory system of the human beings. It invades the circulatory system and cause some endocarditis and also cause some systemic diseases in the human being. So that's why it's a very previous disease, and the study of pathophysiology of this dental caries is at most important. So for these Streptococcus mutants, generally the dental doctors or dental researchers are facing the in vivo animal models case. So why we choose this topic is because some of the Streptococcus species causes a fracture disease in the silkworm, like the Streptococcus faecalis, Streptococcus bombaceus, generally cause the fracture disease. So in that line, we tried the Streptococcus mutants in the silkworm. So the madam also explained in the last speech. So generally we use the animal models for a scientific purpose. There are long-standing practices for production of vaccines, antibiotics, the fundamental understanding of human disease process. Generally we use the animal model. But nowadays approximately 20 million animals are used in the biomedical. So if you use this much of animal models in the biomedical research means definitely the ethical issues are rising. So nowadays taking the ethical concern and ethical committee clearance is a very big issue. So instead of using so many animal models, especially the mammalian models, it's better to use invertebrate animal models in the preliminary stage of research. So it will help to speed up our research and also helps to minimize the usage of mammalian animal models. So to overcome all these limitations, generally some of the scientists are trying to use the C. elegans, Drosophila melanogaster, but showing some little success. So we are coming from the sericulture background. Silkworm is a very good invertebrate animal model system. It possessing so many analogous tissues, organs. It shows similar sensitivity to pathogens, comparable effects of drugs. It's a low cost, no ethical conflict issues, and no longer the poison. So in this manner, we use silkworm as a model animal for Streptococcus mutans. So our objectives of research is Determination of S mutants induced toxicity in the complex memory. Evaluation of phenotypic and biochemical changes due to S mutant infection. To evaluate the implication of these S mutants and medicinal compounds and biological and biosynthetic types of B. So we use the material like Streptococcus mutants and then larvae of 56 silkworm larvae and then antibiotics commonly used against Streptococcus mutants. So these are the methods we followed. And then when it comes to results and discussion, so first we tried whether the Streptococcus mutants induce any death in the silkworms or not. So we inject the Streptococcus mutants into the silkworm system and try to see whether the death is causing or not. So luckily, we injected these Streptococcus mutants in 3 different routes. For example, oral, midgut route, and then intrahemocytic. So in all the 3 routes, the Streptococcus mutants caused death in the silkworm. So, but the LC50 values, concentration required to kill 50% of animal, the concentration required is varying from the route of administration. For example, in the oral route, it requires very high concentration. In the midgut route, it's required 2.1 to the power of 6. When it comes to oral route, hemosil route, it's only 10 to the power of 4 concentration required to kill the 50% of the silkworm larva. So, what are the pathological symptoms we observed in the silkworm larvae after injecting the Streptococcus mutants? So, first one is the control larvae, and second one is the larvae treated with the Streptococcus mutants in the oral route. In the oral route, when we treated with the Streptococcus mutants, one special symptoms we observed, it's damaging the silkworm pouches, the abdominal legs having that pouches. So these Streptococcus mutants were causing some damages in the pouches. So these are the same images of the pouches. These are the control larval pouches, and these are the pouches affected due to the Streptococcus mutants. So, and second one is to the hemosil or a midgut route. Generally, a child like excreta, rectal protrusion in the preliminary stages reduces the food intake. So, all the and the flaccid body, these are the common symptoms what we observe after infection, the Streptococcus mutants. And in the hemolymph color also, when we inject the Streptococcus mutants into the silkworm body, the silkworm larval hemolymph color is completely changed. So this is maybe because of some once the bacteria enter into the silkworm hemolymph, the humoral immunity system is activated. So the fat body generally releases the serine proteases, phenol oxides like enzymes into the system. So it causes a melanization in the film. So it may be one of the reasons to get a turbid of so the hemolymph get converted into turbid. So this is the dissected the Streptococcus mutants infected silkworm larva picture. So here the cell gland, fat body, and then the dorsal blood within the alimentary canal is also completely damaged because of the Streptococcus mutants. So for identification of any bacteria, the PCR is a best method. So we use the PCR amplification to this. Generally for this, we taken the GTFB gene. Generally for identification of Streptococcus mutants, generally they follow the GTFB B, C, and E. GTFB B and C is responsible for production of water insoluble glucons. The glucons helps to adhere the tube surface. So and then GTFB D is responsible for the production of water soluble glucons. So generally we choose the GTFB B gene, and in the except in the control and then PBS injected batches, in all other batches like hemolymph injected and then intramidgut and then in the oral route, we find out Streptococcus mutants in the silkworm body. So once we confirm that these Streptococcus mutants infected the silkworm, we tried the antibody testing in this study. These antibodies generally we choose the advent and toxin. Advent and toxin is generally using against the Streptococcus mutant, the human being. So we tried these advent and toxin. So recovery was noticed in a 2.50 microgram of advent. Then 33.33% of recovery was noticed in the case of advent, and 23.33% was noticed in the toxin. So the low recovery was noticed in the low as well as the higher concentration. So optimal dose of antibodies for the best recovery is a 2.50 and 5.0 microgram per larvae. in both the antibodies. So generally the larval, up to the 15 star spinning rate, the larva is somehow good. Means that the recovery rate is good up to the spinning level. When it comes to spinning level, the larval death is more. Maybe it's due to declined antiviral activity of this abated tax because we do only one dose. So here, after antibiotics treatment, some of the larvae is recovered from the Streptococcus mutans infection and spun the cocoon as well. And we done some SDS path analysis to see the effect of Streptococcus mutans on the hemolymph protein profile. So here, when we inject the Streptococcus mutants mainly affecting the 180 kDa protein, 83 kDa protein, and 29 kDa protein. So 83 kDa protein is generally a storage protein. So when the larva is in the larval stage, generally feed the mulberry leaf and store this protein, and then it's finally used for the metamorphosis. So generally, once we inject the Streptococcus mutants, generally the intake of food is very less, and then larva is more weak. So these protein intensity of this band is very much reduced in Streptococcus mutants infected batches, and then in the amoxicillin or taxine infected batches, again the band intensity again is increased. So here for the oral route, we tried the different doses. For example, in the hemophil route, we tried only one dose. In the oral route, we tried 1 dose, 2 dose, and 3 doses. So here also the recovery rate of the larva is somehow improved to 32.22% to 2.5 microgram per larva, and then it's improved up to the 48.33%. So these are the protein profile. Here also in the third dose, in the 6th day, the larval hemolymph with 83 and 28 kDa proteins intensity was improved. So finally, the silent findings of this research, we successfully demonstrated the settling Streptococcus disease in the silkworm larvae. The P-mori larvae shall be used as an in vivo carology model. Since the clinically advocated antibodies could effectively control the disease set in the silkworm system, the efficacy of the newer molecules that are being established to combat the S. mutans infection shall be evaluated. The effect of S. mutants and antibiotics on the protein profile of the humans explain the molecular basis of evidence. The differential up and down regulation of 83 and 30 kDa proteins due to S. mutant infection and antibody is not only offer the similar studies in the human, also open ample scope to discover the biopeptide drugs against the human dental cancer. So, thank you.
A good study. I'm open for discussion. Any questions? It's okay, please.
Is it Prasad working that by 3D printing or then Prasad? Your work is funded by whom?
Professor.
Correct. Funded by which year? I know, but DBT Bharat project was.
DBT Bharat project is different, sir. They are doing some Saracen based work, Saracen and Python based work.
Bioprinting in capital. Your work is model. That you can apply further. Further work, you have already done what you are only protein. Can you take further? Only protein is just for model only. You cannot have any. You would have done what the other countries would have done the 2D, then you would have done some sort of this.
Applications are there, your findings should reach to the public. Telling it is not just a part, it's not start. A lot of applications are there for the human welfare, or the beneficiaries are much more utilizing this. Let us do it. Can you do this in Devilmata? Is it possible?
No, no problem. If there is a bill, there is a
You have to collaborate.
Our director is always supported to do some research.
Yeah, already I asked to remove his name. I am the message on Golo. And the rest people will be ignored as.
Already sir said, don't sit white in the forms. Better to do some research. I will ready to support.
Because if it's a health perspective, the concept is health perspective. It's a dental health, mental health, physical health, and plant health, insect health, bacterial health, okay, human health, fundamental health, right? Generally, we should do something good.
Actually, we are planning with one mega project with one startup company. The health aspect. Very soon only. It will be one of the.
Yeah, we cannot ignore everything. Everything is important. Even when you see for salt is important. Okay, summer is the water is important. And it is my brother is very, very important.
Next, I invite Dr. Lalita Natarajan Sankhya to talk on influence of post-nutrient assimilation and efficiency on full quality and very simple Samya recipe.
Okay.
Please.
No.
Good afternoon to all. Respected sir, sir, esteemed delegates of this seminar. Now I am here to present the topic on influence of post-nutrient assimilation and efficiency on cocoon quality and reproduction of very silkworm Samia recipe, Donovan, Lepidoptera saturninii. As a children, I wanted to tell, Samia ricini is a multi-voltine, and we all know that it's a domesticated insect just like our mulberry silkworm, and it can be reared on diversified host. And we should also notice that the reproductive phase of any silkworm is dependent on the non-feeding adult stage, which was solely dependent on the food energy accumulation during the larval stage. During the larval stage only it is eating, and during the adult stage it is not taking any food, and the entire performance, reproductive performance is dependent on the larval stage health. So now we should concentrate on the microenvironmental conditions, also the larval health, which is influenced by the host plants which we are feeding during the larval period. The present study is conducted just to understand and reveal the impact of the assimilation rate of different food sources on silk and as well as the egg production of Samia recipe. So assimilation is nothing but the ingestion and digestion, the difference between the ingestion and the excretion. So during the time, the food is present in the gut and it is getting absorbed and the digestion happens. So in between the ingestion and the excretion, the assimilation happens. The assimilation is very important for the larval. So now we are going to talk about that assimilation portion only. So a comparative performance analysis was done in 3 host plants. Mainly, the first primary host plant is the castor or eri, and the secondary host plant tapioca and kessil. Why we have selected tapioca and kessil? Since in Assam, we cannot have castor all through the year. All through the year, castor production is not good like non-traditional states like Gujarat and other places where castor is grown as a main crop. In Assam, it is not possible to maintain castor all through the year. So, eri silkworm being native to Assam, we are in a condition to produce eggs. We are in a condition to produce a lot of seed production, and our targets is also increasing. So now we have to find some alternative sources of food plants which can perform equal to the primary host plants during the winter where the castor crop is not available. So we have done this study to comprehend the silk and egg production where we get the equal as that of the primary host plant. So the experiment was conducted in P2NE basic seed form during October-November, which is the most favorable season. And the eco-race selected for the study is Gurdwara eco-race. And here we have done the full budget analysis of the consumption, excretion and assimilation rate. And it is done as per the Waldberg method, and all the formulas were used. And we have done the energy budget and nutritional indices were calculated, and the data were subjected to statistical analysis. And castor rearing was done, tapioca rearing was done on the variety which was selected from the CTCRI, Central Tuber Research Institute has given some varieties to us for evaluation, out of which Sericulture and Majuli local, these varieties were found to be very good for, and as equal as the primary host. So we have selected the Sericulture variety and used in this study, and Kesaru H0047 variety is found to be the excellent one, so we have used that variety. And the food ingestion pattern was studied in all the 5 stadiums, and digestion is also studied on all the three host plants. And it can be seen that 34 grams of leaf has been ingested by a single larva, and 12 grams of excreta is done. And here 30, and in 3 host plants it is calculated.
Ingestion, huh? So it's addition. Instead of expression, she mentioned ingestion. It's a new word. Ingestion, okay, down.
Digestion, excretion.
It's a word, this is standard word. Have you read any books?
Yes,
That is there.
Yes.
You put it there, first time I am getting.
Yes, sir. It is there, sir. Register. Excreta is also called as register.
Okay.
So the assimilation is calculated as per that ingesta and digester, and 22 grams is getting assimilated, and according to that, tissue weight gain gram per larva was also calculated. And the approximate digestibility for all the stadiums were evaluated, and here we can see that there is no significant difference in the approximate digestibility. However, the 5th insta showed significant variation because it is high in the castor and reduces in the tapioca, followed by tapioca, and next one is the kesaru. And this is the larval duration. Since there is a digestibility difference, we can see there is a 2 days difference. Kesaru will take 21 days for completion of the larval duration, and tapioca 18 days, and it is just 18 days in the castor. And likewise, there is a difference in the larval weight also. And this is the regression pattern. Castor showing the maximum, and there is a very good relation. And this is the food conversion efficiency of the silkworm. Most of the conversion efficiency in larva, cocoon, and shell, it is insignificant. Influence of host plants on the silkworm larvae, cocoon, and egg production parameters was studied. And here we can see that the shell ratio is 15 in the case of castor, and 14 in case of tapioca, and 13 in case of kesaru. The fecundity is an important factor which we were concentrating on. Here we can see that 326 number of eggs were laid in the castor fed larva, and 311 on an average, and 297 in the case of kesaru. Also, this is an important table because with respect to the ingesta and digesta, here we have calculated and found out that there is no significant difference between the host plants as per the amount of quantity of host plants which is fed to the larva. The egg production is not getting affected. And the oogenesis is also proper in all the 3 host plants, and it is on par with the castor. And to summarize, the larval duration is here, and the energy budgeting, we can see that the ingesta and digester required for producing 1 gram of cocoon and 1 gram of egg was insignificant among the 3 foods. So this is very important factor with which we can finalize taking, or we are Sericulture Report is investing money on purchasing the seed cocoon. So if we are purchasing a tapioca rad or a kesaru rad cocoon also for the same price as that of castor, we should ensure that we are getting proper egg laying in that also. So this study is very significant because the cocoons are as equal as that of castor in case of egg production. It can be comparable. However, 1 gram of shell production required for the lowest ingesta is 63 in the case of castor, and 69 in the case of tapioca, and the kesaru is 79. Here I would like to summarize that point, that 10 to 12 kg of fresh leaf biomass is required for rallying one DFL and 240 kg of excreta production for 100 DFL rally. And the way forward is, it is concluded that tapioca and cashew plants are potential alternate hosts and offer comparable yields in any seed cocoon rallying. So we can convince our farmers to go for tapioca rallying in a good variety, particularly in Meghalaya. I am working in Northeast region. So particularly in the Meghalaya region, there is a good productive zone for this seed cocoon production. So there we can promote tapioca and cashew rallying instead of castor if they don't have during the later season and enhance our egg production in our unit. And with this I conclude and thanks for the opportunity given by the organizers.
Introduction. Open for this.
So this is point only cut couple safety. Cut couple one end of open.
Yes.
So you are using this tapioca, queso, and then castor and everything.
Yes.
And recommending the dark is reason. Is there any economically the industry can be more important like that of mulberry cocoa?
This is not related to mulberry, this is related to any from feed production.
Right.
Actually, we have only 2 units in Sericulture for feed production. Now we are promoting agriculture in non-traditional states, apart from Northeast also. So the requirement of seed is increasing and our target is also increasing. So for satisfying the target, we cannot produce the entire seed cocoon requirement in our own farm. So we have to depend on the farmers. The farmers do not have that many castor lots to give me 15 lakh seed cocoons during my requirement. Conduct the rally all through the year. All through the year. Recommendation is that during the winter and during the spring season, in which the castor is not ready for cutting, we have to go for tapioca and the castor varieties to sustain the seed production.
You can switch over.
Castor will not be there, all be yellow, and we have to do the full variant in castor tapioca, which we have standardized in our.
So very good. Very good. Not compare.
One thing I want to ask.
Sir, cashew will feed more.
Yes, 2 days.
2 days. But fertility is less.
Yes.
Number of fertility,
Yes.
number yes. But the number of eggs per gram almost no secret.
Yes, sir.
How do you calculate that?
Number of, sir.
One is larval period more.
Yes, sir.
The expected is more number of eggs.
Yes.
But it's showing less.
No, the larval period is more because the nutritional sufficiency is attained after that 20 days only. But in castor being the primary host, in 18 days itself, the nutritional sufficiency is complete.
Yeah.
But
Once the larval period is more, the expected is more people by.
no,
No. The question is
yes,
that you are because of that only given the larval period more, number of eggs per gram is less.
yes,
But your number eggs per gram. X per gram is no significant within 3. How do you correlate?
correlate means the shell is reducing, but the egg is not reducing. The egg, all the 3 rallies, I am getting above 300 on an average, but the shell weight is reduced. Two cocoons of casero is equal to 1 and 1/4 of the castor. So that much shell difference, shell is that much equivalent, but our egg production is not affected. So this is.
Now, what is going to say? There's a call LCW, larval critical weight. We see the larval critical weight.
No.
We read the literature, maybe 1986 literature. What where one method, another method is that they find the critical weight. Depend upon stage, maybe 15 star, 3rd day, 4th day, recommend for the critical weight. That is substance for this one. Because it depend upon your feed frequency, feed quality, and because assimilation, pH, so many things are, it's a complex process. They have to set the larval critical weight, that will tell This is the days required for the polar spinner. That will correlate with the number of eggs less, number of that will correlate. You see, it's same work only. One more setting make it large physical.
Okay, okay.
It's nobody.
But one observation is, Kesarim is not having that much moisture as the castor.
Okay.
Castor is having lot of moisture, so excretion will be more. A lot of, due to lot of excretion and lot of nutrient absorption, that water content is there. The completion of the larval duration is first. But in Kesaru, it is tight and it is giving good nutrition also. In winter, already the time, it is a time taking process, the duration is extended. But it is not getting affected with the egg production. So we recommend that it can be taken as a good seed. Sir, I thank my team. I also convey my thanks and acknowledgement to my team as well as CSB for allowing me to conduct this. Thank you.
Next question is Dr. Mala and Sil on yield attributes of various samyarism in the rearing conditions of traditional and non-traditional practices.
A very good afternoon to respected chairperson and committee members and my dear delegates. So myself, Dr. Mala, scientist from Central Silk Board. Very fortunate to be a part of this international conference and would like to present a paper on the topic which titled Yield Attributes of Erisilcum samyarismi in the Rearing Conditions of Traditional and Non-Traditional Crops. So as a well-known fact that India stands second next to China in the raw silk production, and India is having this unique of producing 4 different kinds of silk. So where Erisilcum stands second. So as a known fact, Assam is the traditional state for agriculture, and the rearing of silkworm is of heritage and prerogative for the tribes of Northeast India. So now the era has come to agriculture because of the unique features what it is having, and what to say that now to reach the set goals as per the Flint Sumatra target about the 60,000 metric tons. So agriculture plays a pivotal role in increasing the demand because of its unique properties. Like it is highly polyphagous in nature, having both annual and perennial plants, for which it has to be dependent on house plants, and it is tolerant to biotic and abiotic stresses for easy living practices and value addition, as well as it is a employment generating sector and with the low investment and high incomes. So as India is having most non-traditional states like Gujarat, Jharkhand, Chhattisgarh, Karnataka, Tamil Nadu, where gas is grown as a main crop for seed production. So that can be used for growing Erisilcum as well, because 25 to 30% of the leaf foliage can be used for our agriculture without foregoing its economic leaf litter. So with that, there is a scope for horizontal expansion of agriculture. So keeping it with this fact, this study has been designed with an objective of analyzing the Erisilcum performance in traditional and non-traditional crops. So the current state is like around 2.5% increase has been observed from past decades, and 99% of the Erisilcum is produced nowadays from the northeastern India only. And now recent days, because of the impending climatic factors and the seasonal shifts, so Northeast India is facing problem for production of the superior quality of the cocoons. So with this, so there is a scope for horizontal expansion outside of the Northeastern India. So the research methodology adopted was, we have selected 2 centers, P2BS of Kapatoli and ESSPC Hosur for carrying out the research. We have selected 12 farmers, 12 ASRs for our study. Yes.
We consult about the traditional.
Yes.
Also, it's not.
Yes.
I'm sure non-traditional will perform better than.
Yes, that is.
Then,
Yes.
I'm telling we are asking, we non-traditional. You have not studied that. Okay, please.
Sir, we have selected ASRs under these 2 clusters, and we have supplied with this is 3 lanes from ESSPC Hosur only.
And we have done monitoring at every seed crop level, and we have across the seasons we have conducted the study. And meanwhile, the meteorological data has also been documented. And after the completion of the rearing, we have purchased the couples and we have assessed for its formula. And finally, data was compiled and subjected to statistical analysis. So we have studied under 4 seasons. So here we have taken both separately male and female wise for the cocoon shell and shell ratio. So here, as for the cocoon characteristics, so Assam has documented the highest value of 3.34 average of the 4 seasons when compared to the Tamil Nadu area. So it is a similar trend for all the patterns, especially for the cocoon characteristics. But when it comes to yield attitude, the entire scenario has changed. So here we can see that here Assam, across the seasons, we have a significant difference because the seasonal impact was more in Assam because within the day itself, a lot of fluctuation was seen in temperature and relative humidity. But whereas in Tamil Nadu, a minimum and optimum trend was maintained, and there was no significant difference could be noticed within the seasons. And sir, here, sir, crop 1 means it is a June to August. Sir, actually, this September, October, favorable season is a favorable season. Favorable season is a favorable season.
Crop 1.
Sir, crop 1 is uncertain. Sir, it is from June to August. June to August. So these, sir, between the crops, there is a significant difference in Assam, but there is a non-significant difference in Tamil Nadu, showing that this particular Tamil Nadu, there is not much drastic changes in the climatic factors. And with this table, we can show that the C2, that is at reading during September to October, it favors the season for agriculture, both in Assam as well as in Tamil Nadu. So when it comes to the cocoon yield per 100 DFLs, maximum of 62.37 per kg has been observed during the crop 2 season, and the lowest was observed is 19.16 in the crop 1 season in Assam. Because it is a summer season, which has temperature has drastically increased, and that has favored for the lesser cocoon yield. And this is the meteorological data. So a maximum of 39 degrees Celsius it is observed in Assam region, and when it compared to the Tamil Nadu, it is only 33 degrees Celsius. And with the relative humidity of 100% is also noticed with respect to the season. And this particular figure shows that there is a 44% increase in the cocoon yield per 100 DFLs. This shows that agriculture can be potentially taken in non-traditional areas for example, expansion. And meanwhile, the set targets can be reached if we can able to expand this in states like Chhattisgarh, Jharkhand, and Karnataka, Tamil Nadu as well. So, this rearing activity starting from incubation till the harvest of the cocoons, and both the ASAS levels at the P2 of crocodile and ESS PC. And the findings from the study was it is a realistic performance what we have documented at the ASAS level. But when it comes to the farm level, the scenario is changed because under the protected condition we will do. But at ASAS level, this is the significant differences what we have observed in Assam and Tamil Nadu. So unlike Assam, there is no much difference. Drastic fluctuations was observed with respect to climatic factors in Tamil Nadu, which was positive sign for taking up silkworm based enterprise development projects. And irrespective of the crops having different seasons, so silkworm can be reared throughout the year under the agroclimatic conditions of Tamil Nadu. As in the earlier presentation, madam said like during winter and spring season, castor is the main problem in Assam. So we can switch over to the other plantations. But that is not a similar case in Tamil Nadu, where throughout the year we can go for castor rearing. So this is what we have drawn from this study. So thank you for this opportunity. Thank you.
One for discussion, if one or two persons will survive, if how many minutes in between during lunchtime you can interact due to want of time, no? We want to direct chairman advice to complete all the
You have written Assam.
Yes.
For Tamil Nadu, you have written TN. That is not correct.
Tamil Nadu TN we use, here Andhra Pradesh EMP also we use.
Ah, that is standard.
Yeah, standard.
You that's
Standard.
what, yeah, Assam. Then only they make it one full name, another cut short, no, shortcut.
Any lengthy discussion means you can, during lunchtime you can interact with them.
TN people are.
Please, we can continue next. Any.
Prasanta. Oh, fast, it's me.
Very good. Next, Vasant, extraction and analysis of physiochemical properties
Extremely happy.
of oil from very sector. Please restrict your presentation within 5 minutes, please. Because we have some 13%.
We have 13 posters after this.
Totally 15% expense are there.
To complete within time, posters 23 minutes. For oral, it is 5 minutes. We have to finish it very quickly, then only we can have one or two questions, or otherwise we don't find time to interact. Welcome to another present here. Today I am going to present extraction and analysis of the physiochemical properties of oil from various silkworm. Let's move on introduction. Nowadays, the developing countries are utilizing the insect significant role for human nutrition as serve as a edible byproduct. In India, the northeastern parts of states are utilizing the Sericulture insect pupa as a valuable food product. In addition to that, exploration of the silk production. Apart from that, in silkworm pupae contribute 60% of masses in a single cocoon. As per the report said, and annually approximately 23 metric ton available in India. These byproducts we have effectively used to significantly produce the nutrient supplement and the food security. And objective of the research is first we had explored the oil from dried pupa and then to analyze the physiochemical properties of silkworm pupae oil. Let's coming to the methodology. First, we had collected the first pupa, then drying, then powdering, then we have to analyze the two methods. One is substrate method and another one is column extraction method. This method used to extract the oil and further subjected to the physiochemical properties. First one is first we have to collect the fresh cocoon followed by dematuration of airy silkworm pupae. In that we have tried the different time interval. We had isolate the 8th hour dematuration is high compared to the second hours. After 10th hour, the pupa will overdried and it will affect reach to the oil withdrawal. Then coming to the extraction method, in this extraction method, we have tried two methods. One is substrate, another one is column extraction. We use 5 solvents. Among these 5 solvents, we isolate extent, give more recovery compared to the all solvent. In substrate, we observed 22 gram per 100 gram of dry pupa. In column extraction method, we observed 25 gram per 100 gram of dry pupa. It's coming to the physiochemical properties. These are the value observed in the airy silkworm pupae oil, which is on par with the other edible oil like sesame and groundnut oil. Hence, we finally concluded pupae oil is used for soap preparation and cosmetics and other pharmaceutical values. And final, my conclusion is we standardize the time of dematuration of airy silkworm pupae, which is 8 hours, and in column extraction method is higher recovery compared to the substrate extraction method. In physiochemical properties are clearly showed pupae oil used for edible and other pharmaceutical purposes. Thank you.
How many kgs of the pupae required to get 1 liter of oil?
So actually, this is not my sir. Instead of my senior is unfortunately not named there. That's why I'm presenting. I have to go out of this.
People, the easy people,
Yes, sir.
think it is bigger than the
Yes, sir.
mulberry required. 6 kgs of paper is required to get 1 liter of oil. Probably you have 5 kgs because the paper is bigger. It is not your work. But you present it well.
Sir, 30% we can get, sir.
Huh?
30% oil we can get, sir.
30%.
30%.
No, how many kgs of paper required? But and then get 1 liter of oil. 5 to 6. Yes, right.
3 to 4, 5 pages.
You have seen oil?
Yes, sir.
Yes, oil. What type of oil you have seen? Tell me how many types of oil? Well, oil.
You can contact me, sir. Next I will call you for me, sir.
Yeah.
You can present on synergistic effect of honey.
I actually, the presentation starts at 10 o'clock every day. This is the very first time I'm checking all over.
No
I
problem.
change my patient myself, see all of my students are.
Please, sir. Please restrict your time within 5 minutes. After that, 13 more we are having, sir.
You're all exhausted. Okay, it is self-explanatory. A very good afternoon, and I thank the Dr. Mandir Murthy and Dr. Mutulakshmi Madam and Dr. Chideshwari and Dr. Panavellu for all presenting over here for this assessment of the technical session. And today I would like to present the findings of my project group students who have been done in last year. The synergistic effect of the honey and lemon juice enriched mulberry diets are the economic traits of the mulberry silkworm complex. Mulberry silkworm complex is a sole important economically beneficial insect. We should be able to see that the domestication in order to use the economic parameters assessment and see that evaluation of all the characters. And firstly, the improvement of the raw silk quality mainly depends on the nutritional composition of the mulberry and its genotypic expression of physiological and biochemical parameters of the host plant. The honey is a nutritious sweetener and multifactorial nutrient of medicine. It's widely used in the sericulture as an exogenous nutrient in the silkworm mulberry diet. Honey can be solid or liquid, whose color changes from colorless to dark brown depending upon the source of honey, stimulates the feeding and speeds up the absorption of mulberry leaves. It has been demonstrated that the diet is rich in honey and improve the larval development and also ability to produce the dark silk. And secondly, another one is the lemon, which is a member of citrus genus, belongs to the family Rucaceae. Lemon is rich in a vitamin C and includes number of minerals such as potassium, salt, calcium, iron, magnesium, phosphorus, zinc, vitamin A. Supplementing with lemon juice boosts the larval weight, growth, and protein level in the silk. And using this lemon juice and the honey, which are going to help for the enhancement of efficiency, utilization, absorption, and assimilation of all the nutrients, which are very much important for the manifestation of the economic character. Keeping in view that I have taken two objectives in order to investigate what exactly the effect of these two, honey and the lemon juice, and also larval growth, and also the performance of the economic characters from this particular silkworm double hybrid, that is FC1 into FC2. And materials and methods here which have been prepared with three different the percentage of the treatments which have been used during the fourth install in order to see the performance of the economic characters and all the larval parameters. This has been characterized by several scientists previously, and keeping in view that we have been adopted what will be the best source which will be able to facilitate, which will be able to help for the manifestation and improvement of the economic character.
So there have been T0, T1, T2, and T3, and T4. T0 is an absolute control, and T1 is only distilled water, and T2 is untreated 2%, and T3 is lemon 3%, and T5 at 2% honey and 3% lemon juice mixed together. And this is the absolute control where they have chosen the 4 pills of silkworm. And the healthy mulberry leaves have been harvested from the mulberry garden in our department. And that has been used for treatment of this only fresh leaves without any treatment, that is absolute control. And secondly, a distilled water has been used in order to compare with the other treatments. Another one is the honey-fed experimental batches. 2% of honey, 2 ml of honey has been dissolved in the double distilled water and completely homogenized, centrifuged, and we are going to get homogenate, and that has been used for spraying on the lower surface of the mulberry leaves, and which are fed in the 4th instar silkworm. And similarly, lemon juice, it has been directly extracted and prepared 3% lemon juice by using 3 ml of the lemon juice and mix it with the 97 ml of the distilled water and completely mixed and centrifuged together and obtained, and that has been used for the spraying on the lower side of the silkworm, mulberry leaf. And another one is that is P5, where a 2% of honey and 3% of lemon juice fed to the experimental batches, which have been mixed together in order to see that what will be the combined effect which is going to get for the manifestation of the economic. So this is T0, T1, T3, T4, and T5. And these are all the some of the findings which you could be able to see here in the table, where the foliar additives, as I said, larval weight, cocoon weight, shell weight, pupal weight, shell ratio, filament length, filament weight, linear, dendrita, and effective rate of rearing, fecundity, and age. So all the parameters have been considered, consolidated in the FC1 and the FC2. It is a silkworm double hybrid, very popular in the present situation in South India. All the players are going to be interested upon doing this particular breed. And where we could see the honey, which has been used, so that 22.06% of the improvement of the shell weight, and also in the lemon we could see that the 21% of the shell ratio, and as well as the filament length that has been considered to be very high in the absolute control, and also effective rate of silkworm rearing that has been observed in the honey treated batches, that is 84.33%. And fecundity also absolutely increased, and whereas the hatching percentage that has been seen only in the distilled water treated batches. Altogether and consolidated, the honey and the lemon juice, which has been shown the highest linear 2.97 has been manifested in the particular FC1 into FC2 hybrid. When you look at the graphical representation, the larval weight has been observed in the lemon treated batches. It is of single larval weight, that is about 4.84. And the cocoon weight, it has been 2.27, and it has been graphically represented over here. And honey and the lemon are the two components which have separately helping in terms of the larval weight manifestation, cocoon weight manifestation, and followed by the shell weight, where the honey, which has been shown the percentage of 6.12, and the pupal weight is 1.76 in the lemon treated batches, and these are the graphical representation which has been integrated over here. And shell percentage, the honey is about 22.06, and lemon it is about the filament length is 1,522 in terms of meter, and same data has been represented in terms of these two graphs. And filament weight is reflected about 20% of difference, and honey and lemon about the linear is about 1.67%, which has been reflected over here in the form of the histograms. And shell and rendita, it is also important the quantitative parameter, about 10% of the improvement that has been shown, and effective rate of rearing about 84.33% of the effective rate of rearing the FC1 into FC2 hybrid. And this is the one which is reflected in the descending order, that is also a positive trend for the improvement of the economic character. One is that the data is a quantitative parameter. And some of the findings which you could see that at the end, and the couples which were arrested on the different treatment, absolute control, distilled water, honey, lemon, honey and lemon together. So honey and lemon together, which are some more effectively utilized combined effect, synergistic effect, it has been recommended for the utilization of this particular foliar additives. The nutritional impact of honey and lemon juice enriched mulberry diets individually and synergistically, and the numerical growth data and the distilled water experimental batch, honey treated experimental batches, lemon treated experimental batches, and together honey and lemon treated experimental batches compared to the distilled water, we could see that there is a drastic improvement in terms of percentage, especially the larval growth and quantitative and qualitative traits of the symptoms have been discussed. Both the nutrients showed a positive impact on all the economic parameters, but different in the degree of influence. Lemon juice yielded significant gain in the cocoon weight about 17%, and pupil weight about 20%, and silk weight about 3.5%, and linear 11.6%, and lemon juice yielded gain the cell percentage of 20%, and shell protein about 7%, and linear is about minus 11.8%. And the findings of the work that has been done with the conclusion, and the FC1 and the FC2 silkworm larvae fed with mulberry leaves fortified with exogenous nutrient during the fourth instar. We have been taken into the fourth instar separately, fifth instar separately, and I am presenting over here only the fourth instar larvae, which has been chosen for the treatment, not the fifth instar. Wherever you have taken fifth instar, the fourth instar has not done any treatment. You have taken fourth instar and fifth instar not has been treated. And the foliar additives, as I said, 2% honey, 3% lemon, and their combinations were separately prepared and for the spraying in the morning hours to enrich the value of the mulberry leaves. The qualitative and quantitative traits of the FC1 and the FC2 hybrid were assessed subjected to the statistical analysis, replication wise to interpret the data collected after the experimentation. The food additives, honey and lemon and combination plays a dynamic role in the expression of the nutritive values of the mulberry leaves, showed a positive increase with the palatability, digestibility, and the adaptability of the mulberry silkworm during the late instar. The results shows that exogenous additives treated both yield greater fecundity in FC1 and FC2 silkworm hybrid, and results showed that the fourth instar lemon treated batches yield greater larval weight, shell weight in the FC1 and FC2 silkworm rearing was conducted in the department, and treatment wise it was separately for supplementation of foliar additives to enrich the mulberry diet during the fourth instar. So it means to say that the replication wise it has been maintained, rearing has been conducted, and treatment has been maintained in order to enhance the quantitative and the qualitative character, which has been gaps for the synergetic effect or the combined effect in order to see there is any improvement. This is the purpose this experiment has been carried out. We have different mulberry gardens in different seasons. All the nutritive values are not common, but we have to see that wherever the necessary requirements have been added in terms of rearing that has been carried out by our own student, in order to see that a comparative analysis in the different soil, different varieties like B1 variety, G4 variety, S36, S54, and M5 variety. So all the 5 different varieties are there, and finally we have been chosen the B1 variety performing better in terms of expression of economic character, expression of qualitative and quantitative character. In addition to the synergetic effect, we could see that there is a better performance that has been recommended to the farmers. It has been used for the further research in order to see that it's a further study that is required.
Point of interest, it's okay, but how we can use commercially? As an economic point of view.
We can suggest, but they have to procure them.
It will be useful in commercial class, because so much people are free to
Only thing is, you have to make an attempt batch wise. It is not a tough job. You can make a batch wise and see that the manpower has been used and required food additives like honey and lemon that has to be procured, and you can try. Hundred. Yeah.
More than costly, you may need some 1,500 kgs to rare and 100.
No, not like in a huge manner. It is, you have to, replication wise, you have to maintain. That may be recommended.
Okay, sir.
That may be recommended. Huge rating means you cannot do 1,000 lanes of brushing on silicone there. That is very difficult. Labor you can see.
Next.
Thank you.